Chai from the Department of Biomedical Science. I'm happy to have my colleague, Dr. Kamara Ibrahim, who are also from the Department of Biomedical Science as the second speaker of today. Okay, Dr. Kamara, her research interest is investigating the molecular basis of gliomas using multi or mixed approach to better understand the glioblastoma and its implications for future treatment. Hence, today she's going to talk to us about her expertise, okay, with the title of Dissecting Molecular Regulation of Glioblastoma. And if you have any questions about the topic, please leave your questions in the chat box and we will attend to them after the talk. So without further ado, we welcome Dr. Kamaria to start her presentation.
Okay, so for today I'll be talking about my interest on the looking at various aspects of molecular regulation of glioblastoma. And for today's presentation, I will just briefly go through on the basis of glioblastoma regulation. And I'm not going to go into the details of my research. So this is basically a basic presentation on how does glioblastoma works. Okay, so I'm sure everyone knows about, most of you knows about glioma, and there is a, if you see glioma, it's a benign or malignant brain and spinal cord tumor that arises, which can come from glial cells, whether it's astrocytes, oligodendrocytes, epidermal cells, and tumors that arise from astrocytes are then called astrocytic tumors or astrocytomas. Tumors that arise from oligodendrocytes are called oligodendral tumors, and tumors that arise from epidermal cells are called epidermomas. So astrocytes form the main connective tissue of the brain. You can see here, and the oligodendrocytes serve as the specific function of wrapping central nervous system and axons with myelin, and microglial cells exert limited immune function and may have roles in tissue repair and restoration. So neurons are postmitotic cells, and astrocytic and oligodendral cells have limited proliferative capacity. So it is now believed that gliomas are most likely derived from precursors of intrinsic brain cells rather than from dedifferentiated neurons, astrocytes, and oligodendral or microglial cells. So this is, if you can see, this is a representation of glioblastoma taken from imaging, and if you can see here that the origins of glioma, it should it started off with the neural stem cells, and then in normal conditions, these neural stem cells will become neural progenitor and differentiated into neurons, whereas some of them become glial progenitors and become oligodendrocyte or then astrocyte. Okay, so if you see here that there's protein markers for neural stem cells, and progenitor cells and differentiated cells are indicated, and the progenitors and the normal differentiation process in the green arrows originate three main types of cells in the mature central nervous system, including neurons and glial cells, particularly oligodendrocytes, astrocytes, as I mentioned, and epidermal cells, and which are not represented here. The most classical hypothesis on the origin of glioma cells is represented by the orange arrows, as you can see, where it will be arise to become oligodendroglioma, oligoastrocytoma, and astrocytoma. Okay, But the most recent hypothesis postulating that gliomas can originate from direct transformation of neural stem cells or glioprogenitor cells, which is represented in the green arrow. Okay, so the common diagnosis for glioma and particularly glioblastoma is by performing imaging where you use MRI to detect tumor and without and with contrast enhancement. And secondly, histology is being performed, and you can see there's high variable morphologic tumor, and it is termed as glioblastoma multiforme, mentioned forming the pivot of the tumor is fusiform, atypia, and pleomorphic cells, but low-grade neoplastic astrocytes are often detectable more or less. So cellular pleomorphism includes small undifferentiated giant epithelial spindle, gematocytic lipidized and sarcomatoid cells. And some tumors may present one kind of pattern dominantly, and these can be established in different subtypes of GBMs. And another aspect, another essential aspects of diagnosis is by performing molecular classification. And this molecular information is important to achieve greater diagnostic accuracy and much more precise prognosis and optimized patient management care. Right? So this is just a representation of neuroimaging and histological features of glioma. So you can see that glioblastoma, you can see there's a what you call the necrotizing area that is much more prominent. Okay. So earlier glioma classification, it is much more looking towards the inclusion of the molecular classification as well as by looking at the histological aspects. And you can see that it can be divided into IDH wild type gliomas and IDH mutant gliomas, but within that classification, there's a subgroups based on histological classification. Okay, so it is it is very important to determine the molecular markers to tackle appropriate treatment strategies in glioma, particularly in glioblastoma. And you can see that within this molecular classification, the various types of treatment intervention are given to the patient so that a patient would benefit from the treatment. Okay. So there are many altered genes in glioma, and if you can see here that the most altered genes is TP53, and this is common in cancer cells, and then followed by IDH and CDKN2A, P10, EGFR, and so on. So the ones that we are interested in mostly in glioblastoma is by looking at the P10. P10 is another tumor suppressor gene that is commonly mutated or deleted in GBM, of which the loss of P10 function results in the activation of the PI3K-AKT signaling pathway, which promotes cell survival and proliferation. Whereas by looking at the IDH1, isocitrate dehydrogenase, a subset of GBM, in patients who have mutations of IDH1, they would be classified into good prognosis patients. So these mutations are associated with different biological and clinical characteristics compared to more common primary GBM. So primary GBMs, most of them had IDH1 it will not have any IDH1 mutations. Retinoblastoma, retinoblastoma 1 is a tumor suppressor gene involved in cell cycle regulation, and alteration of this RB1 are observed in a significant portion of GBM cases, which contribute to uncontrolled cell division, and the NF1 or neurofibromin 1 mutations in the NF1 gene encodes for the neurofibromin protein. So they are also found in GBM, and NF1 plays an important role in regulating cell growth, and its loss can contribute to tumor development. So these are the genetic alterations genetic alteration based subtypes, where the classifications are based on the IDH mutation, PTEN, and EGFR, which are related to patient survival and can be used as indicators of patient classification. And you can see that from here, the IDH wild type correspond primarily in primary GBM, whereas the IDH mutant mostly in secondary GBM. And in comparison with the IDH wild type versus IDH mutant, you can see that IDH wild type will have more of other genes that has been mutated, for example, third promoter mutation, homologous deletion of CDKN2A, and EGFR alteration, as well as PTEN mutation and TP53. And less of TP50 mutation compared with IDH mutant. But IDH mutant, apart from IDH mutant and TP53 mutations, all genes, most of the genes that has been implicated are less. Okay, so the evolution of GBM classification, it is important that to know that why the classification of GBM has been done thoroughly through these through these 2 decades, is because of the poor prognosis of patients. So each and every year, researchers would do some very much of in-depth work to look on how to tackle the problem of within these patients. So initially, the classification based on molecular has been done by microarrays, started in 2001 by Rickman, and then followed by Han, Nutt et al. In 2003. And after that, with the advance of next generation sequencing or high throughput sequencing, Parsons et al. In the tumor atlas cancer group started off and by looking at how the classification based on classical transcriptome based classification. Okay. And before that, 2 years before that, Philip tried to classify based on the proliferative mesenchymal and neural activity, and followed by Brennan, Verhap in 2010, and Brennan in 2013, where he would look at based on methylation-based classification. And later on in 2016, David and Louis would then suggested that the WHO should have a clear definition on GBM, and they've agreed to classify GBM based on IDH wild type and mutant and OS. Okay, so a new exploration and since then, after that, there's always been a new exploration based on omics and algorithm. Okay, so followed by these other researchers as well. What is interesting or what is important in knowing or catering the proper classification of this patient on GBM patients so that they would be able to get the appropriate treatment. So, for example, here, a study recently by Zhang et al. They've tried to look at whether by utilizing these different classification based on classical, proneural, and mesenchymal based, does the in this case, this is actually a mice study, orthotopic xenograft model, and they've taken the tumor from the patients and profiled the tumors, and then xenografted onto mice and see whether the treatment with temolizumab and radiation therapy would benefit these different classes of groups. Okay, so if you can see here in a proneural group, it will definitely benefit from temolizumab treatment, whereas the classical group would benefit more on for radiation therapy. So this means that we'll be able to personalize the treatment for each patient. Okay, so again, by looking at the classification by Phillips, Verhaag, and Wang, you can see that each individual scientist has their own signatures. For example, Phillips would say that proneural would have a signature of NCAM, GAPBR1, GAPBAR1, and SNAP19, and etc., followed by Verhaag and Wang et al. So basically, the classification suggested are based on molecular or gene signatures. And currently, in 2021, the WHO diagnostic algorithm for diffuse gliomas in adults have been has suggested to be modified, where you can see that all the diffuse glioma will need to be classified into whether the samples or the tissues have IDH1 mutation or not. So, and specifically by looking at the R132H mutation. So, if there is the reason why this mutation is being looked at is because it is much more prominent in GBM. Okay, so if you can see here that IDH1 R132H wild type, if one of the more following in the samples have microvascular proliferation, necrosis, TRP mutation, and EGFR gene amplification, as well as the score of +7 or -10, and you can see that it's a straightforward classification where the tissue will be classified or the disease will be classified as glioblastoma IDH wild type and WHO grade 4. And whereas if it's mutation, then we had to decide whether the mutation, whether it involved ATRX mutation, TP50 mutation, or the ATRX retain and 1p19 co-deletion. And then we would be able to know whether it is classified as astrocytoma IDH mutant or grade 4 or astrocytoma IDH mutant grade 2 or 3. Okay, so I'm not going to go further in that, but basically glioblastoma would fall into the IDH wild type, same as grade 4. So what is the major difference between the previous classification WHO 2016 and WHO 2021 classification is that the classification has been it's sort of like a simplified regardless of the histological morphology. It's a straightforward where you can see here diffuse astrocytoma will be diagnosed as astrocytoma, anaplastic astrocytoma. It's a straightforward. If we classify into whether it's just astrocytoma or glioblastoma, and whether it's IDH mutant or not. Okay, so that's the main thing. And another thing is that the grading itself, the who grade will previously it was in Roman numbers, but now it has been changed to Arabic numbers. Okay, 2, 3, 4. So the major challenges in GBM treatment, although you have a lot of studies that have been done, and a lot of fancy drugs that have been in the market, still the survival of this patient would not fall between around 14 months and more. So it is a challenge because most of the tumors in the brain are localized, and it has inherent resistance to conventional therapy, and the brain itself has limited capacity to repair, and also the ability of this migration of malignant cells into adjacent brain tissue, and the variably disrupted tumor blood supplies, which inhibit effective drug delivery. And also you can find that there is a lot of tumor capillary leakage, which results in a lot of accumulation of fluid around the tumor, and also there's presence of intracranial hypertensions. And as a result, some of these patients would have neurotoxicity due to the treatment that has been directed to the tumors. So the regulatory mechanism of glioblastoma is divided into a few categories: cell migration and evasion, the ability of the cells to proliferate and survive, angiogenesis, the alteration of metabolic activity, immunomodulation, and resistance to therapy. And firstly, we'll look at the metabolic alteration, the glioma nutrient uptake and utilization in the brain tissue context, the biochemical environment and metabolic interaction between brain cells contribute to nutrient availability. So you can see that within here, the blood-brain barrier, and there's a lot of blood supplies and nutrients that allows to the nutrient to be supplied to the glioma cells. Okay, so and because of that, the blood-brain barrier, which is formed by specialized brain endothelial cells, leads to a biochemical environment for gliomas that diffuse, that differs from those of other cancer types, and there's a specialized architecture to unique tumor microenvironment. So in this case, there's the alteration occurs because of the IDH gene. Okay, so the IDH was supposed to provide what you call a metabolic regulatory mechanism to the cells, and it is used. So the glioma cells opportunistically take up nutrients from the extracellular environment, including glucose, acetate, and glutamine, as well as other amino acids, and they use nutrients for energy, biomass production by fueling glycolysis, and as well as all other this TCA cycle, the Oxford cycle, phosphorylation, as well as the pentose phosphate pathway, and this will then increase metabolic activity of proliferating glioma cells to generate what you call reactive oxygen species as byproducts that necessitate the antioxidant production, including the production of reduced glutathione and other molecules as well. Okay, so you can see that why does the IDH mutant respond better than the IDH wild type GBM cells is because that the mutant, the IDH mutant is much more sensitive towards treatment, okay, because of the vulnerability of the GBM genome itself, and you can see in IDH1 wild type, because of the intact activity of the enzyme itself, and also it is able to stabilize the hypoxic condition and allow cell differentiation. Thus, in this group of patients, the treatment or the patients would not respond much to the treatment intervention given. So you can see that there's a huge difference between IDH1 wild type, which has a 14-month survival, compared with mutant IDH1 around 42 months. Okay, right. So there is a, and as I mentioned before, in gliomas, it indicates if the patients have IDH mutation, it indicates this patient has good prognosis, and improved tumor response towards chemotherapy or irradiation in clinical trials. So if you look at the integrative analysis, define glioblastoma core pathways, these are the pathways that allows GBM cells to proliferate and survive. And you can see that the most pathway involved are the RTK, RAS, PI3K signaling pathways, P53 pathway, and RB signaling pathway that has been altered. And also this also shows the landscape of GBM pathways alteration in patient samples, cross patient samples. Okay, so the one of the way that the tumors are able to, GBM is a bit tricky to be treated is because there's an intratumoral heterogeneity, as well as and as well as the intertumoral heterogeneity which occurs within the GBM cells. Also, there's a perivascular niche for neural and brain tumor stem-like cells. So within this niche, the cells are direct contact with brain microvessels that provide specific perivascular niche, and this interaction would allow the astrocyte neurons and pericyte supplies this specialized microenvironment through cell-cell interaction, and which includes the notch ligand, for example, and the secretion of soluble factors which allow the survival of for example, non-stem glioma cells, which then provides tumorigenesis. Okay, so mentioned about intertumoral heterogeneity, and also that GBM cells are able to invade and migrate using their unique way of movement. Okay, so it started with the detachment of invading cells from the primary tumor mass, and then later there's involvement of attachment of the invading cells to the extracellular matrix, followed by the degradation of ECM, and then it has a unique altered cell motility and contractility. And these ephrin receptors activate several pathways involved in glioblastoma invasion. Okay, so angiogenesis as well allows that the GBM cells to survive, and also there's an immune escape mechanism where that these glioblastoma cells has unique representation that is able to escape from the immune modulation, immunomodulation, and specialized tumor-associated microbia and macrophage that is present within the unique lateral ventricle, coral plexus, arachnoid villus, superior sagittal sinus, and so on. Okay, so and the blood-brain barriers and blood-brain tumor barriers also interacts with many uh, immune cells, and there's inflammatory, anti-inflammatory reactions and pro-inflammatory reactions occurs within this unique structure. And because of the time,
Sorry,
yeah,
Dr. Kamara, because it's almost 9, so probably you want to
yeah, this
wrap.
wrap-up
Yeah,
session. So
yeah.
finally, and these GBM cells have temolazumide chemo resistance and resistance to radiotherapy. And what embeds in the future of GBM therapy is that how do we actually tackle the blood-brain barrier? Because the blood-brain barrier is a protective barrier of central nervous system that limits the delivery of drugs to the brain, parenchyma, and the sensitivity of the brain to the therapeutic effect. So and now, because of the apparent nanotechnologies enable drug delivery to be delivered across the BBB hold great promise for the treatment of GBM. As you can see, there's a whole range of organic nanoparticles and inorganic nanoparticles, as well as these virus-based systems and cell-based vehicles. So I think and by understanding the regulatory mechanism of across the blood-brain barrier, it is hoped that in future we will be able to provide the most suitable drugs to to cater glioblastoma therapy. Thank you very much.
Thank you very much, Dr. Kabara, for the insightful and detailed talk. So since there is no questions from the audience, so I guess we shall wrap up the Breakfast Health webinar session today. And thank you very much, everyone, for staying with us for the past 1 hour and wishing you a great start of the day. Thank you so much.
Thank you so much, Dr. Chai. Thank you so much, everyone.